So my protein for expression is ferritin. Starting with the problem statement, since ferritin is an iron storing protein, its deficiency can lead to anemia, heart failure, and various other health problems that are being listed below. Now, what is ferritin? It is an iron storage protein, as I said. Its primary role is iron sequestration. It makes the iron available for critical cellular processes while protecting the lipids, DNA, and protein from potentially toxic effects of iron. And these are the four main biological roles of ferritin: iron storage, iron transport and release, protection against oxidative stress, acting as a diagnostic marker. Now coming to the structure, it is a spherical protein complex. It is composed of 24 subunits, and these subunits are assembled into a spherical shaped structure with the 4-3-2 symmetry, also known as the octahedral symmetry. Now coming to the cloning strategy for ferritin, the gene source is the cDNA of Drosophila melanogaster, which is also known as fruit fly. Here we take our DNA, we splice it, we get the mRNA, and then we do reverse transcription, and then we get the cDNA. The vector used is the H-ferritin. Since there are two types in this, the heavy ferritin and the light ferritin, I selected the heavy ferritin because this vector was able to generate cell lines with significantly higher expression levels, and that is what we ultimately want for our protein. And these two are the primers that I used. Now that we have our gene, vector, and primers, we then do the PCR, that is the polymerase chain reaction, and it is used for the amplification of proteins. And the materials that I used are the DNA sequence, the primers, the Taq polymerase, that is the enzyme that links nucleotides together to form the PCR product, the deoxynucleoside triphosphate that acts as the building block of new DNA strands, and the buffers which provide a suitable chemical environment for the activity of DNA polymerase. Now, how the PCR is done, there are basically three steps. The first is the denaturing, which is the breaking of bonds of the protein structure. This happens at 95 degrees Celsius. The second is the annealing, which is the application of heat to the template to separate double-stranded DNA into two single strands. And third is synthesizing, which is done at 72 degrees Celsius. Here, new strands of DNA are formed. And since I use two restriction enzymes, that is the EcoR1 and the SAL1, so there's a need for the DNA digestion, and this is the protocol which needs to be followed. And here we can use commercially available buffers known as the NE buffer. It is used to reduce large fragments into smaller fragments, and it is also used to increase the stability. Then DNA digestion and ligation, and these are the materials which are being used: the PCR product, the vector, the restriction enzymes, that is the EcoR1 and the SAL1, and the DNA ligase, which is the T4 DNA ligase. And the reason we use the T4 DNA ligase is because it is one of the most versatile DNA ligase, and it can ligate both blunt and cohesive ended double-stranded DNA fragments, and it also helps in stabilizing of the DNA. Now, in the ligation part, the restriction enzymes EcoR1 and SAL1 digests both PCR product and the vector, and then we receive the complementary sequences, and then we ligate the PCR product into the vector using the T4 DNA ligase. We generally do gene ligation because it physically joins the DNA vector to the gene of interest. And then we do the verification of PCR product. We confirm the product is of the correct size by the agarose gel. And the reason we use the agarose gel is because it is easier to pour, the samples can be recovered, and the gel does not denature the samples. The next step is the transformation, and here there are two types that can be done: chemical component cells and electroporation. Here I used the E. coli cells, that is the BLD21. It is because they produce high levels of protein, And the reason I use chemically component cell method is because the results are more predictable, we don't require special apparatus to deliver charge, and we can increase the volume of DNA if the concentration is low. And the major reason of not choosing electroporation is that due to the high electric current, it can cause the cell death, and we do not want that. And these are the procedures that can be followed for transformation. The cell culture and screening. So the plate cultured cells with kinamycin is allowed to form colonies overnight, and then the blue and white screening is done where several colonies expressing clone are picked up. So if it's a positive clone, it will have DH5 alpha plus clone, and it will form white colonies. And if it is a negative clone, that is just DH5 alpha, it will form the blue colonies. Now coming to the protein expression, these are the steps for the protein expression, and one of the important substance that is being used here is the IPTG, which allows for the control inducible expression of gene of interest. So by adding IPTG to the growth medium, we can regulate the timing and the level of the protein of the expression. Next is the tag protein purification and tag cleavage. So tag cleavage is done to removal of the tag, because sometimes what happens is the tag can interfere with the structure and the function of the protein, and the tag cleavage is always performed on the affinity column prior to the further purification steps. This is the procedure for protein purification, and here an important substance is imidazole that competes with his tag for binding to the metal charge resin, and thus is used for elution of the protein from an IMAC column. It can also reduce the binding of contaminated protein, which would increase the final purity. The characterization and verification. So here the proteins can be analyzed after separation by using 12% SDS page. One of the most common is the staining by Cosme Brilliant Blue, and it is a protein specific dye. These dyes bind to the protein in the gel, making them visible as colored bands or spots. And the reason for using Comasi Brilliant dye is because it is easy to use, it has high sensitivity, it doesn't chemically modify proteins, and also it is cost efficient. Now coming to the last part, that is the activity assay. So activity assay are the technology that helps researcher to identify the unique protein expression or the expression patterns. And so the activity assay I used is the enzyme linked immunoassay, that is known as ELISA. And the reason I chose this is because it is highly sensitive. It can detect the low concentrations of protein. It is very specific because it can distinguish between similar molecules. It is accurate than most of the other assays, and it is very easy to use. And this is the principle of ELISA. And the intensity of signal is directly proportional to the amount of ferritin in the sample, and the intensity is measured at 450 nm. And these are the future directions where ferritin can be used, that is, can be utilized in coronary artery diseases, it can be used as a gelatin target, it can be used in cell transplant, and it can also be used in breast cancer.